To appreciate just how incredible this packaging system is, we have to look at the exact physics and structural hierarchy the body uses. If you were to simply throw a thread-like 2-meter-long molecule into a microscopic bucket, it would instantly knot up and become completely unusable. The cell solves this by using a strict, multi-layered, hierarchical folding system that shrinks the DNA's length by a factor of about 10,000 to 100,000 times. The 5 levels of DNA packaging. 1. The bare double helix. Width 2 nanometers. At the absolute baseline, DNA exists as the classic double helix discovered by Watson and Crick. It consists of 2 sugar phosphate backbones running in opposite directions, connected by rungs of nitrogenous bases A, T, C, G. At this stage, it is incredibly long, highly flexible, and highly vulnerable to breaking. 2. The nucleosome. Width 11 nanometers. To stabilize the strand and prevent tangling, the cell introduces specialized, positively charged proteins called histones. Because DNA has a negative electrical charge due to its phosphate groups, it tightly wraps around these positive histones like a rope around a pulley. Exactly 147 base pairs of DNA wrap around a core of 8 histone proteins about 1.65 times. This creates a unit called a nucleosome. Under an electron microscope, this looks exactly like beads spaced out on a string. 3. The chromatin fiber, 30 nanometers wide. The beads on a string structure is still too loose. Next, a 5th type of histone called H1 acts as a clip, securing the wrapped DNA in place. The nucleosomes then coil tightly around one another in a helical or zigzag pattern. This packs the beads close together into a dense fiber that is 30 nanometers wide, shortening the length of the strand significantly. 4. Looped domains, 300 to 700 nanometers wide. During the normal life of a cell interphase, the 30 nanometer fiber is folded into massive loops. These loops are anchored to a central scaffolding of non-histone proteins inside the nucleus. This stage is crucial because it keeps specific, frequently used genes accessible to the cellular machinery that reads data, while keeping unused genes tucked away. 5. The metaphase chromosome, 1,400 nanometers wide. When a cell prepares to divide, it needs to move its DNA safely without tearing it. The looped domains undergo a final extreme level of supercoiling and folding. This compresses the chromatin into the highly visible, dense X-shaped structures called chromosomes. The scale comparison and analogy. Because nanometers and micrometers are hard to visualize, let's scale the whole system up to human dimensions. Component actual size scaled up equivalent. Imagine trying to pack 2,000 kilometers of kite string into a single small bedroom so perfectly that you can pull out any random section instantly, read a sentence printed on it, and slide it back in without a single knot. That is what your cells do every single second. Why this perfect packaging matters.